We are going to treat a little bit difficult topic today. Not really as hard as it is, but because it's got a series of processes to go through that you need to understand instead of memorizing them. And it is one of these topics that bridge clinical biochemistry with hematology. What are those topics that exist or going to be detailed in this lesson? You've got glycolysis, Krebs cycle, you've got glutathione and GSPD deficiency. Now, in the human body, the body must undergo a metabolic process of breaking down the glucose to release ATP. So glycolysis in itself is an anaerobic process that produces the derivative into an aerobic process. The final result is the production of pyruvate. This pyruvate undergoes an aerobic process within the human cell in the mitochondrial matrix. And this step is what we call the Krebs cycle or the tricarboxylic cycle. Let's go away with this one because I just want you to have a clue what the whole process is. But the relevant aspect is what I'm going to teach you. By the way, you can subscribe to this channel and like it as we go on. But the relevant aspect is the structure that I'm going to explain to you. Pay attention to this.
Now, here we go. So, like I was talking about, we are just going to go straight directly onto the structures in the glycolysis pathway. And just to give you a heads up on that, we are going to use diagrams entirely, and you're not going to grab anything, you're not going to memorize anything. You will just follow the structures as we name the different glucose structures that comes along and the enzymes involved and the metabolic process as in energy dissipation and electron transfer, and that will give you a very fair understanding of the whole thing without having to memorize them. So for instance, we all know the benzene structure. So this is a benzene structure. And for instance, I'm going to draw a chain around it. So I'll give this a chain around it, and I'm going to give this to be maybe a chloride ion attached, and that's our chloride ion attached. And of course that we know we can attach certain side chains to give benzene a particular parent name. For instance, if we add this OH group in the form of water to a benzene structure, we change the benzene name to a phenyl. And of course, if there is any CH attached to this chloride benzene structure too, we can have a toluene. So there are different ways we can bring a structure up here, and we can deduce a name for this as well. Let's take this one up here, and then let's deduce a name for our structure that we have found now. So when we select the whole structure on here and we look for a name for this particular structure, we can just, instead of us wasting the time on there, we can just deduce a name for this structure, and this is 3 or bracket 6 chlorohexyl 2-methyl-phenyl. So like I told you, we've added a phenyl structure and there is a methyl structure, and there's a whole long chain of here that has a chloride ion attached to this long chain, and we are not interested in this. This is just a pickup. But more interesting here that we can also look at is that this is the structural appearance, but there is a 3D. For instance, here there is a CH2 that we are not seeing, as well as here and there, and on here you can see a CH that we are not seeing, but there is a 3D structure that we can pick up from here. If you want to see the 3D structure, we can just have a look and see how it appears, and on here we can just move this one around and see this is actually how the whole compound that we have produced looks like. So let's follow suit and see how the glycolysis process is going to be. This is a lovely time. Okay, I will take all these off the screen because we don't need them, and now we go directly onto forming our glycolysis product, which is the pyruvate. Now let's begin. It begins with a glucose. If we know glucose is a pyranose, pyranose means it has 6 atoms in a hexagonal cyclic structure, but one of those 6 atoms is an oxygen. Though the glucose has 6 carbons, but it doesn't have all the carbons in the ring. It has 5 of them in the ring together with an oxygen. So now we can just begin. So we have 1 carbon here, 1 carbon here, another carbon here, and then this in the ring, and we will have 1 up here outside the ring. And we also need an oxygen, which is in the form of water around here. But of course, we have a hydroxyl group binding to this, 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 and then the rest are hydrogen ions which are binding to each other, and we can just go on that. So let's link our structure. And then of course on this side the glucose is having a side chain up here, and that is in the form of alcohol. It's a hydroxyl methyl side chain up here. So we will bind this and link this to that. So this is the hydroxyl methyl, and we have to show the entire structure of the our compound in the form of its carbons present in here. So what I'll do is that I'll select the whole of our structure and I will go on there for structural properties, and let's see what we have produced. 1 second on our atoms. That carbon. Sure. Great. So this is how it appears. So every carbon on here is showing up just as it's supposed to be, and this is the oxygen forming part of the ring on here. So this is our glucose structure, and it's a monosaccharide. So of course, if you want to take this through a glycolysis, you have a present hexokinase. And note, the enzymes are really relevant. When you say kinase, it phosphorylates, or it facilitates the transfer of a phosphate group. So we will just show up here that we have a reaction going on to this direction, and of course we are going to write down the enzymes and any other thing that is required for this process. So the first stage of glycolysis, of course, we need hexokinase. Hexo means 6 and kinase means to phosphorylate or to transfer a phosphate group to hexokinase on there. And of course there is an ATP that's being used here in the first stage of the glycolysis. So this is here, we have hexokinase and an ATP being used up here. So from hexokinase we'll convert this monosaccharide, which is a glucose, to a glucose 6 phosphate. The reason is that it is going to add a phosphate group to this alcohol side chain at the cis carbon. So if we are to name the carbons up here, let me take these reactions out of the cursor. So this is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, this is the fifth carbon. That forms this ring, and the last one will be an oxygen. But outside this ring, if this is this fifth carbon, the next one will be the cis carbon. So this is the cis carbon whose hydroxyl group in this form of this alcohol is going to be replaced by a phosphate group with a facilitator, an enzyme called the hexokinase. So what we're going to do is that we have to just copy the whole structure on here. So we are going to copy this whole structure because it's the same glucose structure on here which is going to go. So I will do Ctrl C and I'll come and paste it here, Ctrl V, and of course when I lay it all down here, the structure will come up here. Great. So what this enzyme is going to do, a hexokinase, is to add a phosphate group to this structure. So what we are just going to make a change to it is just to bring a phosphate group and bind to that. That's the whole thing here. So when we say a hexokinase is going to phosphorylate this monosaccharide, this is what's going to happen. So I will just go straight forward and click on a phosphate group. Let me get a phosphate group attached. Great thing. So I'll just come and paste the phosphate group here, and all that I have to do is just to bind them together. But in binding them, they have to make sense on here. The reason why I'm saying things have to make sense on here is because remember, when we were adding the hydroxyl group to this methyl chain, it was in a form of water. Great. So what that we can do is just to take this one out and bring the water up here, because this is just a hydroxyl group here. So let's bring it outside. This is in a form of water, so we can just get an oxygen on here in a form of water and place it up here. So all that we are going to do is that we have to take this one off, and yeah, here we are free. So now we're going to link them all together and we are good to go. So all that we have to do is to link the phosphate group to this, and now to this, to our methyl group. Now we are here. Of course we have the phosphate group attached to our glucose structure, and then the function of the hexokinase is complete. This structure is what we called glucose 6 phosphate, because this is a glucose structure that has got this phosphate attached to the cis carbon. This makes sense. So we can just write a name down here. Yes, so we've got the glucose 6 phosphate, and we do put a hyphen in here just for the sake of science. Great. So we've got glucose 6 phosphate structure up here, and here we can also write the name of the glucose down here as well, so that we don't get confused. This is just the glucose, which is a monosaccharide, and this is the beginning of the glycolysis. This is the product that begins glycolysis. That is it. So we can go back on to for this discussion, but before then I want to give you this head up. We added this phosphate group. The reason why the process adds this phosphate group to the glucose structure in the course of the process is that, one, the more you add phosphate group to a structure, the more energy that will be obtained in the end, because we want this structure to be quite stable and throughput in terms of the outcome. And then the other reason is that phosphate here is more electronegative, so introduce a negative charge into the system. And remember, the phospholipid bilayer of all human cells is also made of phosphate as well, meaning they are going to repair each other, not accepting this glucose structure from leaving the cell, because of course we need glucose to come out and in of the cells. That's why there is glucose channel in there regulating it. But once there is an extra group added to the glucose structure, it means that it's not going to easily leave the cell, because it will be repaired by the phospholipid bilayer, and it's also not going to be recognized by the transport channels. And of course the phosphate group also makes this glucose more reactive to its divisions and reactivities in the glycolysis pathway, making it easier to produce the latter energy. Okay, so now that we've obtained glucose 6-phosphate, this is the structure. So the next thing is that we want to add more phosphate onto the structure. But remember, just as this side, going back here, we had an hydroxymethyl side chain on which we were able to add the phosphate group to the hydroxyl group on here. We can find the same structure on the opposite side of the oxygen. This is the oxygen, the left side on here. This is the oxygen, the right side on here. This is the CH on here, and it's also attached to a hydroxyl group, don't forget it. So all that we are going to do on here, the next step is the isomerization, and the enzymes here is the phosphoglucoisomerase. What it does, just like we know in isotopes and isomerization, that a particular compound can have the same carbon and hydrogen chains and oxygen chains, that's the same atoms, number of atoms within it, but the structural orientations will be different. That is how we call two compounds to be isomers. It means they are the same compound but different structural appearance. So all we're going to do to glucose 6-phosphate is to isomerize it, to give it a different structure which can have a different name, but the compounds within it and the whole oxygen and carbon and hydrogen and phosphate present will also be the same. So now let's go. Because we know it is just an isomer, we are just going to copy the whole structure up here. Make sure we cover the entire here. Yes, and we do Ctrl C. We've copied it here. What we are seeing is a 3D appearance of the structure that we have up there. So because it's just isomerization just means that you're trying to give it a different shape or different presentation. We have now pasted it here. We can go on our rotation and rotate these structures, yeah, just to give it a new look. That is it. Yes, so just to give it a new look, and we can just bring it down in a way. Yes, so we can just move it. Yeah, now we have moved it down here. Let's look at the structure. We've got the phosphate here, everything and everything. The next thing to do on the isomerization is just to create a side chain, which will be the hydroxymethyl here, which means that we just want this side chain not to be connected to this oxygen, so that it can be free for an attachment of a phosphate group. And therefore we need to break this chain and connect this oxygen to this methyl group instead of that. That's all that we are going to do. So now let's go. Great. This is an oxygen that has got an H that is reacting with this carbon, another H reacting with this carbon. So it is actually an O with two H. Now this and that, we've got this here. So we want to break this up. We now take that one away because we are replacing it with this one. So here we break that O out. So we come up here, we connect these two, and instead of this and that, now because of isomerization, we are connecting it to this carbon to make this structure so free. So this is what we call an isomerization. Let's go back just for you to see it the second time. Great. We had this structure already. We call it glucose 6-phosphate. we want to isomerize it. The reason for the isomerization is to get a side chain which is hydroxymethyl like we had previously. So all that we are doing is to just get water which has the same presentation as this O. We create it here, and this one we break it off. This is the isomerization, the phosphoglucoisomerase. This is what it's actually going to do. We break the oxygen out. Now we are connecting this carbon to this, and then this to this carbon instead of this carbon. Now what we are seeing on here is lovely. It is a side chain created here with a hydroxyl group like we had here before. Here, like we had here before, even before we started the glycolysis process. This is what we've got here, and this is the whole reason for the presence of the enzyme. Now the only thing we are going to do is like a dress up, rearrange the structure to look so nice. This whole structure is the same as this with different structural orientation, and here we were having a glucose on here, but instead we are now having a fructose. So we would name this structure as fructose 6-phosphate, and the enzyme that catalyzed the process between here and there as the phosphoglucoisomerase. Now let's quickly write the name on there and we move to the next step so quickly, and I believe you are just getting it on an ISO and not just by imagination. Quickly, let's get okay. So now we can just do a quick zoom in, and I think it's a bit okay now. Now this is what we've got to. Great. If you look at this structure, it's telling you the next step. If you have got these hydroxymethyl side chain up here, it means that it is likely that we are going to phosphorylate it again, going to add a phosphate group to this structure, and then we will need a kinase. So always know that if it is an isomerase, a structural orientation, if it is a kinase, it's going to phosphorylate, or it's going to catalyze the transfer of a phosphate group either in or out. Whichever way it goes, it will be a kinase that will act on them. So we will need a kinase on this fructose 6-phosphate. We call it fructose 6-phosphate because of course the phosphate group is still on the cis carbon, and we are going to add the phosphate group to the first carbon up here. So now we will say this structure is fructose 6-phosphate, so the enzyme will be phosphofructokinase. It's very straightforward because the kinase is going to add the phosphate group to the fructose 6-phosphate. So now let's go on there, add a phosphate group to this structure, which will be on the first carbon, and let's see the results up here. You can just have a look. This is quite straightforward. We've done it before. We just go in there, select our phosphate group, which is this one, place it on here. Now good. But what we are going to do is that we already know that this O has an H already on this side, and there is another H that is binding to this methyl group. So we will select it in a form of water and place it here as a water, because we are going to take this out. There is one H here and there is one H here, which make it a 2H. So when we take this one out, we can you undo it. Whenever you touch on the single bond with this chain, it adds another bond to it. So this is the actual structure that we have here. We are going to break this one out. So we just take this OH out of the reaction, and now our reaction will be on a methyl group binding to this, and then this binding to that. So you add this phenyl group attached to this, and this now attached to that. Now we have a phosphate group binding to this first carbon, and a phosphate group binding to this cis carbon, and this is on the first and this is on the 6th. So we call it a 1,6 biphosphofructose. And just not to get lost, we know this is the resource going here from the fructose 6-phosphate. So what we will do quickly is just to copy that one to the next step and bring this reaction to where it was before. So I've copied this, copy them, come back here, paste them. Great. And I will just come here, and this is the 3D appearance. When I leave it, it will just drop it down here. But remember, here we just attach that one, so we can remove that. And it was like this before. I love that. So it was like this before, and this is the results that we have obtained. So we know this is fructose 6-phosphate, and we know there is a catalyzed reaction on here that produced that result. So we will bring it quickly here and we draw this line straight forward. That's phosphofructokinase. Great. And remember, we did use the ATP, that's why we added the phosphate group to it, and we say it's okay. So this reaction is phosphofructokinase catalyzing the transfer of ATP onto this hydroxyl group of the first carbon on the fructose 6-phosphate to produce the 6-biphospho-fructose, like I've written up here. Great, so we are on here, but just to give you a head up about this that I haven't talked about yet, this is ATP. It is adenosine triphosphate, meaning it has an adenosine attached to 3 phosphates. And remember that we are just catalyzing the release of a phosphate group with the help of this kinase for a glycolytic pathway, meaning only 1 phosphate will be used here and there will be 2 phosphate left here. So it will be adenosine diphosphate remaining after this reaction. Not only on that one, but remember that up here where hexokinase is catalyzing the conversion of glucose to glucose 6-phosphate, this hexokinase also used a phosphate group from ATP, so the remaining here will also be ADP, adenosine diphosphate. Now fast forward, we go straight down to our structure on here. If you look, it's like a spider with 2 hands, 1 hand here, 1 hand there, and the head here, and then the feet down there. But interestingly, it's not a spider, but it's named 1,6-biphospho-fructose. So what we are going to do, the next step, is to catalyze the division of this into 2 equal parts, 2 isomers, which are interconverting. So this is what we would need: an aldolase. An aldolase is an enzyme that is championed in dividing compounds into 2. What this aldolase is going to do is to break this into a glyceraldehyde 3-phosphate and a dihydroacetone phosphate. Now let's quickly divide it. Let's see this process. I'll go down here, and because we are going to use the same structure, instead I'll copy it down and we work on it like we did earlier. So this reaction will come up this way. Let me use, yes. So now we have aldolase. Now aldolase is just alone, not going to use any ATP to catalyze this. So you will have aldolase on here, but I think we can just get this one drawn and rotate. Okay, structure. We tend to rotate it a bit so that it will look the way we want it to look. Good. And that's it. Let's take it a bit back. Let's take this a bit back. Good. So that is what we are actually looking for. And let's go and copy. Good. So this is what we want to copy and bring it down here. And please pay attention to this relevant reaction on here. Now we've come, Ctrl V, we've pasted it, we have moved it close to what we want. We've got all our reactions on here. Nice one. So we will rearrange this structure to look exactly the way we want it to look, and we are going to divide it into 2. So now let's see. The principle of this reaction is such that glyceraldehyde 3-phosphate will automatically, because it has the aldehyde at the end, C double bond O, would have to be automatically attached because the aldolase is undergoing a retro-aldo cleavage, which will automatically bind a double bond of oxygen to this carbon, and therefore this oxygen will automatically be added to this dihydroxyacetone phosphate. That's why it is also called dihydroxyacetone phosphate, because it automatically obtains this acetone to its side. So let's put on our mind that on this right side we are obtaining the glyceraldehyde 3-phosphate, and on the left side we are going to obtain the dihydroxyacetone phosphate. These two are the same. That is why they are called isomers, and they can be interconverted against each other. That is, we can convert one from glyceraldehyde 3-phosphate to dihydroxyacetone phosphate, and then the dihydroxyacetone phosphate can also be converted back to glyceraldehyde 3-phosphate. But the body's mechanism accepts the structural orientation of the glyceraldehyde 3-phosphate. Therefore, the DHAP will be converted back to G3P, that's the glyceraldehyde 3-phosphate, and it will now continue with the glycolytic process. I hope this really makes sense, but what is interested here is how to get the reactions present. Now let's move. like we know before, this is an oxygen, and technically it is binding to this carbon with a hydrogen, binding to this carbon with a hydrogen, and therefore it is an oxygen with 2 hydrogens, which means water can represent this reaction. And we are automatically adding this O onto the left side, which is the DHAP, dihydroxyacetone phosphate, and therefore we will break this bond. And now we've broken this bond. Now let's bring this water a bit closer to where we want to form the bonding. So now you connect this to that. This is what you've got, and you want to create the double bond on here. That is why we've called this an acetone. So now the double bond, which was a breaking of the oxygen from these two sides, have now been added to only this carbon. The next thing is how to break this bond as well. Now we are on there, we break that bond. And that's exactly what the aldolase is going to do. But remember that the aldolase is a triose sugar and it has 3 carbons. So it is automatically going to add a carbonyl group to make this glyceraldehyde 3-phosphate. So we can just quickly do that with addition of water here, and then that, and I'll just connect this to that, straightforward, and I'll do that. Great. So that is it. So we've got 2 sides of these structures. That's the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Let me quickly separate them, write the reaction on there, and we go straight to the next step. And as well, we are going to see the following process. Let me quickly separate them. Good, so that's what I actually wanted to bring on board. So this triose phosphate isomerase catalyzes the conversion between the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate, and both of them can be interconverted. But the necessary one needed at the moment to continue with the glycolytic pathway is the glyceraldehyde 3-phosphate, and it means that our next step will focus on the glyceraldehyde 3-phosphate. And don't forget this, that the glyceraldehyde 3-phosphate is a biologically recognized intermediate, and it can be interconverted into different angles. It can be converted back to dihydroxyacetone phosphate, and this glyceraldehyde 3-phosphate is necessary in the glycolytic pathway because in diabetes, because of the inability to regulate the glucose, the energy tends to come from different angles. The body tries to solicit for this deficit, and therefore it uses other sorts of metabolic products. For instance, it uses amino acids, lactate glycerol, to convert it into glyceraldehyde 3-phosphate, which continues with a metabolic process. So you can understand that glyceraldehyde 3-phosphate is generally recognized, and it's a biologically recognized thing. And even in plants, in the Calvin cycle in photosynthesis, glyceraldehyde 3-phosphate is biologically recognized for photosynthesis. And those are just by the way. Our next step is for us to be able to add a phosphate group, but the idea you should have about this glycolysis is that the end result is for the body to obtain more energy in the form of ATP. So the body will now continue in a process where it can obtain more ATPs. So it adds an inorganic phosphate. So remember, the previous stages where a kinase was processing a phosphorylation was the addition of organic phosphate from the ATP energy. So now this step will have an inorganic phosphate being added to this carbon, and of course this hydroxy group has to make way, so it will be removed by a dehydrogenase, and therefore the enzyme that's going to catalyze this process is called the glyceraldehyde 3-phosphate dehydrogenase. And you have to copy this structure on here because it's exactly the same. And of course we are going to use a P and a small I, which means it's an inorganic phosphate, not ATP. No. And I say, okay, it's still coming up. Okay, yeah. So the size is now smaller. Anyway, good. So we can have this one on here and remove our structure here. a bit backward to make space for our senior enzymes. Good. So let's rearrange this to make a very good space. Yeah, so now we've arranged the structures just to make a very good space for our reactions on here. What we are going to do next is to make sure that we have our inorganic phosphate added to this glyceride 3-phosphate. So let's go on there. This is the structure, and we'll get our inorganic phosphate added. But the thing is, we are coming to add it to the first carbon here. The first carbon has got a hydroxyl group, and of course there's one H and another H reacting with this carbon, so definitely it's in the form of water, which we can put up here. And of course we are coming to add our inorganic phosphate that we can populate the structure up here, which is this phosphate, and we drop it here. So the relevant aspect to follow next for this is to get rid of this hydroxyl group, and to do that we can just come on here and we just get rid of that. And of course, if you want to do our reactions, and this that we have produced is a phosphate group on the first carbon, phosphate group on the third carbon. So this is carbon 1, 2 and 3. So this is 1,3 biphosphoglycerate, and that's the exact name that we can give it to it, because 1 phosphate on the first carbon and the third carbon. This is a very high energy intermediate of glycolysis, and even in the Calvin cycle, like I told you before, in photosynthesis in plants. And we should remember that it is not only this, but it is twice of this. The reason being that this is glyceride 3-phosphate and this is dihydroxyacetone phosphate, which will be converted back into this. So a double of this that is going through this process. So don't be confused when we get to the energy dissipation. And this is the high energy level that is for ATP production during glycolysis. So we will continue with the next step to follow. So you've got 1,3 biphosphoglycerate. The interesting thing here is that the next steps that will follow this stage will all be ATP production steps, and these are where the cells get their energies in the form of ATP. And in this step we will see that there will be energy released due to the presence of these phosphate groups. So we would we need an enzyme that is able to phosphorylate the transfer of a phosphate group, and these are the kinases, and the product here is 1,3 biphosphoglycerate. So the enzyme required for catalyzing these steps is called the phosphoglycerate kinases. It's so easier to mention the names. So what we will quickly do is that we have to select this whole structure and copy it, because it is the same thing that we will use down here. Now when you look at this structure, I've already told you the enzyme that is present here for the catalyzation, the name of the enzyme. I'm going to write on here quickly. And what you should also know about this step of the glycolysis pathway is that because we are going to do a removal of a phosphate group from this structure, there need to be an ADP, that is adenosine diphosphate, which will receive the phosphate group to form the ATP. So in this phosphoglycerate kinase, it will come along with ADP. How many molecules? 2 molecules. The reason why we are going to use 2 molecules is not because we are just removing only 1 phosphate to form 2 ATPs, but because I've told you that on this step where the aldolase divided the structure into 2 for us to get the dihydroxyacetone phosphate, all of them will be converted back into glyceride 3-phosphate, and therefore what we are working on here is a double of that. So there will be 2 ADPs that we are going to add on to this structure, and quickly I will type here as ADP. Great, now we are back here. We have to get rid of this because it is the process that used 2 moles of ADP with the help of this kinase, and we produce 2 ATPs. So you also write down here the ATPs that we will also produce, and then let's get rid of this one. So what we are going to do now is to get rid of this phosphate, and that's it. So if you want to test yourself to know that the structures that I am generating here are correct, I can just automatically generate the scientific name for this structure, and you would type on your Google to see if it is exactly where we are. I will just select the whole of this structure. Let me just go on here to this and that, and I think I can select through this, and I'll go on my tools and I'll say generate the name of structure that I've got on here, and it will come up here. So I will not delete this one, I'll just keep it down here. But if we are to name this structure, because we've taken away the phosphate group from the first carbon and the phosphate group on the third carbon is still linked, this will be 3-phosphoglycerate. Interesting. So we will type our name on here as 3-phosphoglycerate, and of course if you search on Google 2-hydroxy-3-phosphonoxypropanoic acid, it should correlate with this 3-phosphoglycerate, because this is the extended name, scientific IUPAC. This is just the common name that's known on here. Yes, we will go forward with the rest of the structures and the energy dissipation. Don't forget that we are gaining 2 molecules of ATPs on here by releasing 1 phosphate, because we got a double of this structure because of these transitions. Good. So don't forget we got ATP. Yes, and I've written it down here that we've got 2 ATP generated from the process of converting this stage onto that stage with the use of a kinase for phosphorylation or a transfer of the phosphate group. Yes, so don't forget our next step, we are still on the ATP production business. We already discussed that from this stage coming on here will be the production of ATP. So we would have to rearrange this structure in order to make it easier for an enzyme to phosphorylate this phosphate group, or to transfer this phosphate group to other ADP to make an ATP. But transferring a phosphate group from a third carbon is quite difficult. So there should be a rearrangement of this structure so that the phosphate group can come closer on the lower carbons, which is the carbon 2. And in this case, it's like what is properly known in science as mutation, because when we say about mutation, it is either a relocation, a translocation, a deletion, a transfer, something similar to a structural change. And this one is it's not just an isomer, but a breaking up of one side chain from a third carbon to a second carbon. So let's quickly draw this one and show how we would be able to transfer the phosphate group nearer. But what is the enzyme required for this? About the enzymes, like I was talking about, if you look on the preceding stage, we have a phosphoglycerate kinase, and here is the same phosphoglycerate, but is it a kinase? No, we are not transferring a phosphate group, but we are rearranging the structure, which makes it a mutase, like I explained quickly. So this is a phosphoglycerate mutase converting this stage of the reaction, and I'll quickly bring up the reaction up here. Yay, yes, that's drawn here. This is the reaction, and what I'm going to do is that you know that this is just the rearrangement, so I'll copy this one. The whole thing we are going to do is to break this phosphate group and bring it here. So we can just first select our phosphate group, which is this structure already that we are on, and then we will paste it here. And let's rotate it for it to face our structure, making it a bit easier for us to do our reactions. You just rotate it like that. Lovely. So here we are just going to break this phosphate group off. We've broken it off, and we are going to react this phosphate group. And of course, whenever we have a phosphate group reacting in such a manner, we should make sure that it is reacting not to this carbon, but it reacts with oxygen. That's why it is always written as OPO3 on there. So what we are doing here is that, so you see O is reacting with phosphate, O with that, and then it's left with one that could react with that. So it is the oxygen rather reacting it. So we pick up a water, and then we put it on here. We take this one out, and we react this with the phosphate group, and then the remaining with this carbon of our interest. So the mutase has done its job to move the phosphate group from this third carbon to the second carbon, and what you do is that you can just easily rearrange these positions to make it look quite appealing. Here we haven't changed anything. So we now have the phosphate group on the second carbon, and it used to be 3-phosphoglycerate, because the phosphate group was on the 3rd carbon, now it's on the 2nd, so it is now a 2-phosphoglycerate. This name is quite straightforward. Let me write it on here, and here we go. Now we've got 2-phosphoglycerate. On the 9th stage of the glycolysis pathway, it's interesting, it is the 9th stage, and the enzyme there is called enolase. And on the 9th stage, let's look at something. This structure that we've got, the hydroxyl group here, hydroxyl group, hydroxyl group, hydroxyl group ending up here, which actually looks like it is water all over. So this stage enolase is responsible on the 9th stage of the glycolytic pathway to remove water from these 2-phosphoglycerate. So on our next step, it's a way that we will show that there is water remover and there is an enzyme called enolase acting on them. And I'll put up this enzymatic reaction bar on here and remove. Yes, so of course we've got these enzymes here and water being removed from where? From this structure, from 2-phosphoglycerate. So we know that it is this 2-phosphoglycerate that we will need, so we will copy it and then come and paste it down here. Now you've got this, I'll paste it here. So we are going to remove water from the structure. From where? From this 3rd carbon here. So this water will definitely, this will go away. But when it goes away, what else can we see? Now when it goes away, a double bond will be formed here. So this water will be removed and a double bond will be formed between these 2 carbons. And look, this is how it will be. We have removed this one and then we want to create a double bond here. So this is how the double bond will come up. We will create a double bond here, and now we can see this is what we have found as the new structure. And what is the name of this? It is called the phosphoenol pyruvate, and I'll write down the name of it here as phosphoenol pyruvate. Now we are on our last stage of the glycolysis cycle, and of course on our last stage we are hoping to remove energy for cellular activities, and we are going to get energy in the form of ATP out of it. How does it occur? Of course, anytime energy is formed, there is this phosphate group removed to add up to an ADP that forms the ATP. So now they would need a kinase, as we have discussed earlier. So the kinase that will work on here will be this phosphoenol pyruvate kinase, and it is shortly called the pyruvate kinase because it is the kinase that is going to phosphorylate or cause the removal of this phosphate group from this preceding structure onto an ADP to form an ATP. So let me quickly draw the structure up here and we will discuss it further. And of course, yes, I've written in this reaction line the pyruvate kinase enzyme required to catalyze this process. And of course, these 2 moles of ADP will be required to receive 2 phosphate group to form 2 molecules of ATP. That's why I've written here as 2 ATP will be produced. So of course, let's quickly select this because it's definitely going to be our next structure to edit on here. So I'll create this one and that's it. So you will select this and I'll copy. And of course I'll paste on this side as well. So I'll say now paste. So it is now pasted here. So I'll just let it rest. So what we are going to do here is that now this is the results from here. So this pyruvate kinase is the enzyme that catalyzed the transfer of a phosphate group to these 2 molecules of ADPs to form ATP. And it's not like one phosphate being transferred to 2 molecules of ADP. It is 2 phosphate because previously we've talked about it that there is 2 glyceraldehyde 3-phosphate molecules undergoing this process. So you transfer the phosphate group onto ADP and you form 2 molecules of ATP, which means that this phosphate is no more available because it has been transferred into energy production. So let's get this off our process and the remaining structure will come up. But how do we get rid of that? The most important thing to note is that we are getting rid of this phosphate. So when we go on here, we can catalyze the breakdown of this, and when that is done, it means that this particular structure phosphate is gone. Now let's understand the basis of chemistry here. In the enol form of pyruvate, which is the phosphoenol pyruvate, for the reason of thermodynamic stability, that is for either conjugation or substitution, it is preferred that the double bond would exist between the carbon-carbon chain. Now, on this other side, it is not in the enol form. The substitution that occurs here is that it is preferred for the double bond to exist between a carbon and an oxygen. And therefore, this double bond will be broken and the double bond will come up here. So what we are going to do is to facilitate this reaction that I have just explained. Now we want a single bond here and a double bond here. Great. So we have established this basic. Let's see if we are being brilliant on our explanations. The only confirmation is that we would have to select this and generate a scientific name for it, and whatever comes up, type on Google and check if we are thinking. All right, so this is what we're going to do. Quickly, I'll select this reaction, and I've just done that. And the next thing is just to generate the name. Tools, generate name of structure. So you have 2-oxopropanoic acid. Now let me type 2-oxopropanoic acid. What search engine is that? 2-oxopropanoic acid. Still thinking. Okay, 2-oxopropanoic acid, commonly known as the pyruvic acid. Great, so we've got our answer sorted, and we know we've done the right thing, so we can close this page and come back here, and we'll write the name of our structure as pyruvic acid. So because we are so glad that we've got the right structure on here, we will just enlarge this on here and bring it a bit closer to this side, and we will find to type our name here as pyruvic acid. Great work. So for now, what we can do is just to rearrange our structures and give it a good shape and color. So for instance, you can say we'll give our blue color to the names of the products and red color to enzymes. So I will just go on there and select the red color, and I think this should be a red color now. For instance, this, and we apply color and see if it will work. Good. This is also enzyme, like okay, machine. Good. Yes, so beautiful. Now I think we've added enough beauty to our structures, and this is what we have obtained. That's a great work. I'll copy this on here, Control O, and I'll say copy, and I'll paste on a word document. Control V. It took a long way to get us on here. So this is our final work on a Word document. However, I just want to draw your attention to this, that we did not count the ATPs that were produced. But just quickly, if we look at here at the hexokinase, we lost 2 ATPs. So this is our first lost, not again lost. And then on our second part, where phosphofructokinase, we dissipated ATP into producing this, so we lost another ATP. So fast track down here, where we produced these 2 glyceride dihydrophosphate and dihydroxyacetone phosphate, which can be transferred in between, or which can be converted in between. So it means from this stage, everywhere that we take this glyceride dihydrophosphate, all our products will be twice of it. So here begins ATP production, and then here we have 2 ATPs produced because we understand it from this side. So now we have produced 2. We have cancelled out the 2 that we lost. So we lost 2 previously. We've produced these 2. We've cancelled it out. So even though we've produced 2 here, but we are saying it is a loss because we have already used 2 ATPs. And then let's see if we will produce any other ATPs. Of course, on this side we produce 2 ATPs. So in general, we are producing 4 ATPs, but the net is 2 because we used 2 of them during the process of glycolysis.